So we're gonna get started. So thanks for coming. So this is part of our kind of CVE faculty spotlight. The idea is that a lot of our faculty go out all over the world and talk about their research, but then we often don't get to hear them explain those things. So we get an opportunity to hear kind of from one of our own, Todd Przybycien. So Todd went to undergrad at probably one of the best undergraduate institutions in the world, Washington University in St. Louis. That's because I also went there. He did chemical engineering and chemistry there. And then he did his graduate degrees, master's and PhD at Caltech. And then worked at Monsanto for a few years before coming to academia to RPI. We lost him for a few years at another school where he started a biomedical engineering department. But then we brought him back, so we're really happy to have him back. Todd has a bunch of awards, including fellow from a bunch of societies like AICHE and ACS. He also works in a pretty wide spectrum of chemical engineering, from drug delivery and things like Marangoni flows and surfactants and all things like that. But what we're going to hear about today is some of his work in kind of bioprocess design in the quest for $10 per gram mAbs. So without further ado, love to hear from you, Todd.
Thank you, Patrick, and thank you all for having me this morning. It's a pleasure to share my quest with you. Let me tell you a little bit about where we're going to go today. All right, very good. So, monoclonal antibodies. I realize that not everybody wakes up in the morning thinking about monoclonal antibodies and how you might supply the world's demand for monoclonal antibodies. Let me share some of that with you. Wanted to give you a brief introduction to monoclonal antibodies. I wanted to talk about manufacturing processes for biological materials and biotherapeutics. And we'll come around to the so-called platform process, the current manufacturing process for monoclonal antibodies. And then I'll talk about an alternative that we're developing, and I'll wrap up for you. So, maybe it's 2 a.m. in the morning. You're a little blurry-eyed, you've watched the 10th rerun of Real Housewives of Waterville, and you're just trying to figure out what's going on. This woman comes on the television. She'd like to sell you something. She'd like to ask you to ask your doctor if Humira might be right for you, or maybe Cosentyx, or maybe Taltz, or Remicade, or Opdivo, or any number of different drugs. And let me tell you, your doctor can't wait for you to tell him or her what drug you should be prescribed. They stay up all night waiting for your advice. Anyways, there's a common theme here, and that's mAb. Trade names are up above, the drug names down below. They all end in mAb. These are all monoclonal antibodies. Monoclonal antibodies, huge and important class of biotech drug, and it's the one that we're going to focus on today. It's a Y-shaped molecule, monoclonal antibody. It's about 150,000 molecular weight. This molecule is a linked dimer. It's got a heavy chain and a light chain. The heavy chain is shown in this here, and up here. But most of this structure, this 150,000 molecular weight material, is common for all the different molecules I showed in the previous slide and all the hundreds of different antibodies that are available or in development. What is different between each one are at the tips. This FV, the fraction variable region, the very tips are where the specific binding activity of an antibody is exerted. And so those vary. But most of the rest of the structure is common. And this is a class of antibody, part of the IgG class of antibodies, That's circulating in our bloodstream. It's one of multiple different classes of antibodies, but is the most common. It's the one that's most heavily present. And you have thousands of these circulating in your blood at any given time, thousands of different types of IgG class antibodies. What makes this a monoclonal antibody is now in the drug, every molecule has the same tip, right? So it's targeting the same species. It's an important class of therapeutic, as I mentioned. It targets typically cancers and inflammatory diseases. It exerts its effect through the specific binding. It may bind some circulating signaling molecule and take it out of action. It may bind to a cell surface receptor and turn that receptor off and block it. It may bind to the receptor and tickle it and make it exert its own action. So there are a number of different modes of action that the specific binding can be harnessed. There are a number of new targets for antibodies, including high cholesterol, infectious disease. Right? During the pandemic, there were applications to treat those who are already infected with antibodies. Also, Alzheimer's disease. Biogen's taking a lot of shot on goals here, and if they have a very successful anti-Alzheimer's molecule, this is going to be huge in terms of volume. The volumes of these new targets are at least 10 times the size of the market volumes for cancers and anti-inflammatories. In terms of approvals, there's about 173 different mAbs that are approved by the US FDA. The European equivalent, the European Medicines Agency, EMA, 114 therapeutics. The pipeline is full of new antibodies waiting for approval and under development. The value in 2024, $263 billion. If you saw $263 billion lying in the street, we bend over and pick it up, right? It's a lot. So it's a good-sized market. Compound annual growth rate, almost 15% projected. When I look back through the years, because I've worked with antibodies for a number of years, these projections are made all the time because it's such an important class of molecule and the market is so huge. There's lots of market studies. It always outperforms these projections. The demand is incredible and the number of opportunities is huge. So there's a lot of focus on monoclonal antibodies. All right, let's talk about biomanufacturing. How are these going to be made? This is where we come in. Here's a generic view of biomanufacturing. You'll have some raw materials that come in to process. You'll have an upstream process where you have a biocatalyst. The biocatalyst could be cells, could be a collection of enzymes. You'll produce some crude product. That crude product needs to be purified. If it's a biotherapeutic that we're making, this is something that may be injected or maybe even administered in pill form or patch or something like that. It's going into your body, so it has to have a high degree of purity. So there's some purification processes that comprise a downstream process. This entails use of more raw materials and generation of wastes. And then we'll come up with bulk drug substance. This is the purified material. It's not ready for administration, but it's purified, right? Highly purified. This bulk drug substance then goes through a formulation and delivery or fill and formulation operation to get it into the delivery form that will be administered to patients. It might be a solution in a vial. It might be a powder within a vial that needs to be reconstituted. It might be a syringe. It might be a pill. Number of people in the department work on bioprocessing, right? If we were to talk about making mRNA, for example, then we would use a collection of enzymes. This would be an in vitro transcription operation. And so this is an area where John Dornick, myself, Steve Kramer, Jordan Belfort, Pankaj Karan work in these kinds of processes, in particular on the purification. After this has been made by IVT, how do you clean it up and make your bulk drug substance? If you're talking about other types of therapeutics, we might have, instead of a collection of enzymes, we might have a cell line here. If we focus specifically on monoclonal antibodies, we're talking about Chinese hamster ovary cells, and they're going to secrete this antibody into solution. We'll do a separation of the cells from the surrounding fluid, the supernatant, and have harvested cell culture fluids, HCCF. That's the raw feed into the downstream process. It will have several rounds of purification that happen here because we're not going to be able to do the purification in one step, and the purity requirements are really high, so we have a number of things to do. The first major purification is known as the capture step, and then there's cleanup that needs to be done. These are polishing purification steps. Both types are important. When we're done here, then we'll have monoclonal antibody or mAb drug substance, and that'll then proceed to formulation and fill operations to produce mAb drug products. Again, using raw materials and generating waste as we go. All right. We're going to focus on the downstream process today, on the capture and polish purification. That's where my expertise lies. Monoclonal antibodies have been around for a long time. And as I mentioned, of that 150,000 molecular weight, most of that is common, antibody to antibody, despite the fact that there's 173 different approved monoclonal antibodies. Common structure, just the tips pretty much vary. That means when they're produced by CHO cells and secreted into the solution, the material that results is pretty similar, drug to drug. So the downstream process, the purification process, is pretty similar as well. There are certainly differences, antibody to antibody, where things have to be tweaked for optimum performance. But by and large, the same approach is used. This has given rise to the so-called platform process. And this has been known for quite a while. If we look here, this slide indicating the platform process from 2010. And I'll point out Abhinav Shukla is one of Steve Kramer's alumni from his PhD group. So this process, here's the upstream. We have a series of steps to grow up cells to appropriate volumes and then ever larger bioreactors in a train to produce material for the production bioreactor. Some harvesting operations to do the cell separation from supernatant, and then we get some crude harvested material. What does that look like? Will be anywhere from 1 to 15 grams per liter. 1 to 3 was pretty common, maybe 5, 10 years ago. Now we're seeing reports that are significantly higher, maybe 10 grams per liter, maybe up to 15 grams per liter. We have a number of contaminants that we have to remove. These drugs are made by cells, right? But we don't want to break the cells open if we can help it, because the drug is secreted, the antibody is secreted. If we break the cells open, then we have to deal with all the cellular contents. Nonetheless, mammalian cells, like Chinese hamster ovary cells, are kind of fragile. They do break and still are guts, so we've got to do some separation here and remove some cellular material. So we'll have low molecular weight species, lots of host cell proteins, fragments of mAbs. We'll have high molecular weight species. These tend to be protein complexes with DNA, chromatin, also aggregates of antibodies. We'll have host cell proteins on the order of, say, 100,000 to 500,000 parts per million, maybe higher in some cases. We'll have host cell DNA, because again, some of the cells break open in this process, even though we'd rather not. That'll be on the order of 10,000 to 100,000 parts per million or more. We may have some virus particles. We have to pay attention to that. These are mammalian cells. We're mammalians as well, right? So if mammalian cells have virus, the potential exists that we could pick up some of these viruses. They're not removed. Typical virus titers, somewhere between 10 to the third and 10 to the ninth per mil. Virus is particularly tricky, right? Because one virus particle can ruin your whole day. So that's a difficult separation. That's the upstream. That's the feed to this downstream platform process. So there's an affinity chromatography step using protein A. Protein A is a molecule that was evolved by Staphylococcus aureus to bind our antibodies. So we get infected by Staph A, a staph infection. What the antibody does is grab, or what the infecting organism does is grab antibodies and decorate its surface with our own antibodies. It kind of hides behind that shrubbery and says, I'm not really here, don't pay attention to me. So we have identified this specific binding molecule and harnessed it for separation purposes. It's very highly specific, it's a strong binder, so the whole idea is that you pass your mixture through this column, it has protein A in it, the antibody sticks to protein A, nothing else does, and you're all done, right? One-step purification. That's the ideal world. The real world is like, it works really well, but there's still more purification to do. And then there'll be a couple chromatography steps, polishing chromatography steps to remove residual materials. Again, removing aggregates, host cell proteins, and other species that the protein A didn't get to. And again, because the structure of antibodies is common, this can be used pretty much for every antibody. The part that protein A recognizes is in the common part that's shared among all the IgG molecules. Where you might see some differences in tweaking is the specific conditions or maybe some changes in some of the resins that are used here to do the final cleanup. And there are a couple of other steps, filtration, virus removal, etc. Where do we end up with here? So I show the feed material coming in, harvested cell culture fluids. The protein A does a great job in cleaning things up. We'll get a little bit of concentration because it's a bind release type chromatography. We'll knock the low molecular weight and the high molecular weight species down. The host cell proteins in particular knock down to somewhere on the order of 100 to thousands of parts per million. Big step down in terms of that contaminant level. DNA, huge step down, typically down to about 10 parts per million. Virus removal is reckoned in orders of magnitude, so 1 to 3 orders of magnitude removal or log removal value. And one thing we have to worry about is that a little bit of the protein A leaches off the column. And that protein A, unfortunately, is something that generates a fever response in humans. So one of the drawbacks of using protein A is you get a little bit of this material that leaches off that has to be removed later. By the time we get to bulk drug substance, we're on the order of maybe 100 grams per liter. There'll be some concentration steps here. Low molecular weight and high molecular weight species down below 1%. Host cell proteins on the order of 10 parts per million. DNA on the order of 10 parts per billion. We'll have a total of 16 to 20 orders of magnitude removal of virus. Does that mean 0 virus? Hopefully. And protein A removed down to the level of at least 1 part per million or lower. This process, again, dates from this description here from 2010, and the platform was developed a few years before that, and it's been described in great detail. This is 2026, right? It's been known for a really long time. Yep. So, so what's the problem, right? It's all good, man, right? The problem is that we've got some challenges. Satisfying the world's demand, all right? So it turns out that protein A is the bottleneck. This is an expensive chromatography step, so it's the bottleneck in terms of cost. It's the bottleneck in terms of raw material usage. It's another recombinant protein that has to be produced by yet another biotech process, right? So there's a supply chain issue here as well. It's very expensive, only supplanted recently by the affinity chromatography media that's used to purify mRNA. Very expensive media. And it's the throughput bottleneck. It is a bind and release chromatography mode, and it's run inherently in a batch type operation. Now, there's been a lot of clever work. Again, more than $250 billion market. So people are thinking about this all the time and working to refine and optimize. So there are periodic continuous modes of operation that approximate a continuous operation that makes more efficient use of this media and does help to relieve some of the bottlenecks and cost issues, but it's still the bottleneck in all these respects. All right, let's think about now sustainability. When I think about sustainability, I had my eyes opened by this textbook by Heinzel, Beaver, and Pooney, Development of Sustainable Bioprocesses. Dates from about 2007. And I teach periodically, not periodically continuously, but periodically a bioprocess design course. And for several years, I used this particular text. It's still great, but 2007, things have moved on, and there's new things to talk about. But what it opened my eyes to are the different ways that sustainability can be thought of and should be thought of. There's the economic sustainability. I mean, everybody, when you mention sustainability, immediately goes to environmental, which is important. But if things aren't economically sustainable, it doesn't matter if it's environmentally sustainable, yes or no, because it's not going to be done, right? So economic sustainability, we measure that by something known as cost of goods, right? The cost of manufacture. And current cost of manufacture could be as low as $50 per gram of mAb. Probably more typical, something between $100 and $200. It's a little hard to get a fix on this because companies don't put this on the forehead of their employees, right? The cost of manufacturing. It's certainly low. What are these sold for? Tens of thousands, 100,000s per gram. There's a lot of other costs there that are being borne by the sales price. There are other sustainability metrics for economics, right? Net present value, return on investment, and other metrics. We're going to focus on cost of goods. There's an environmental component for sure. And one way of measuring the environmental impact of manufacturing is process mass intensity. How much raw material is needed per unit of product produced? In this case, how many grams of raw materials per gram of mAbs? Typically about 10,000. 10 kilograms of raw materials to make 1 gram of product. Yeah. Think about that. Think about the demand. Right? A lot of this is water. Okay, so well, what's the big deal with that? All right, water everywhere? Not quite, right? Now, even thinking about the Bay Area, California, manufacture, it's tough. There are water use restrictions. And this is not just any water. This is high purity water. So there's raw water that has to be purified at a high level first. So there's some fraction of raw water that becomes high purity water. That's part of this, kind of the 4 grams per gram mAb. There are other metrics here in the environmental sustainability, greenhouse gas generation, for example, CO2 in particular, and other metrics that we could talk about. I'm going to focus primarily on process gas intensity. And the final layer of this is social sustainability, right? Global access. We have trouble paying for and supplying the demand for mAbs in the Western world. And this is at, the sales prices that are, pretty extreme. Even if these drugs were to be sold at the cost of goods, that's not inexpensive enough for the rest of the world to be able to afford these. The Western world has a great deal of difficulty affording these drugs. The rest of the world, right? It's only a very thin cross-section of society that's having access to these drugs. So we've got a lot to do. We've got to knock down the cost of goods. And another aspect of global access is making enough. We make enough to satisfy what we can afford in the Western world. Again, but the demand, these diseases are global diseases, particularly if we start talking about pandemic disease, where that tells you that we've got to be worried about this, because even selfish, self-interest says pandemic disease, we've got to treat everywhere, because if there's pockets of disease anywhere, nobody's safe. So we've got a lot of demand to meet, and capacity throughput is an issue. During COVID, it was discovered that IL-6 levels, interleukin-6 levels in the bloodstream were elevated, part of an inflammatory response. Of course, the lungs were inflamed, right, in COVID patients. There's an antibody that is an anti-IL-6 antibody. It's sold by Genentech, Actemra. And so that was harnessed to treat patients that were infected, to help try to head off this inflammation and help them get off of these breathing assist devices earlier. The fact that this material Actemra had to be diverted to COVID patients meant that the rheumatoid arthritis and cancer patients that were using this drug had access. Right? So that's a really tough decision to make. We've got to be able to get around that. So this is what's motivating our work, trying to address the sustainability aspect. All right, well, so what do? I mean, there's this process that's been in place for many, many years now, and it's been published many times over and over. If somebody walked in the door today and said, Todd, I have a brand new antibody, how are we going to process it? You could point to 1,000 papers in the literature and say, follow this recipe basically and tweak it, and you're going to have purified antibody. That's not good enough, right, for the sustainability goals that we want to get. We got to think differently. One of the things that motivated me here was some even older bioprocessing work coming out of E.J. Cohn's lab at Harvard. So at the outset and during World War II, all right, there was, of course, a lot of demand for blood products. How do you ship blood products out to the battlefield? Well, you really don't, right? You can't ship whole blood out there. Nowadays, perhaps, but back in the day, that was going to be a problem. So what Kahn figured out is, well, look, we can take blood and we can fractionate it down to its useful components, and then maybe freeze dry those components and ship that out, and then it can be reconstituted wherever it's needed and be used. And so he had to think of a way to separate the components of blood. And he came up with the contractionation process. So it's a series of operations that's used to fractionate the proteinaceous components of blood. And in our circulating, in our blood, right, we have anywhere between 5 and 15 grams per liter of IgG class antibodies. Looks a lot like HCCF. Can we do the same kind of thing, right? A simpler approach. In his approach, he used a series of different pHs and ethanol concentrations to generate different precipitates. So starting at high pH and low ethanol concentration, able to get a first fraction, pull that aside, change the conditions a little bit, drop the pH, up the ethanol concentration, precipitate out yet another class of protein, tweak, and he had a number of different pathways to generate these different fractions that he proposed, and this is 1946, to do this fractionation. This kind of thing is still done today by the plasma fractionation industry. It's augmented by some much newer technology, chromatographic technology, to help get higher purity levels, but there are still precipitation fractionation steps in here. So we took this to heart and thought, you know, is there an approach here that we can use? And yes, there is. So this is what we're thinking about here. Across the top in these boxes is the platform process. Could be batch, it could be continuous or semi-continuous. Some capture step that's based on protein A. We have a viral inactivation, a couple polishing steps for removing residual contaminants, more viral filtration, some conditioning, formulation, and fill to get us between drug substance and drug product. Can we replace this capture step with a precipitation-based capture step? And the idea here is that we would purposely precipitate the antibodies, leaving everything else behind, using, being inspired by E.J. Cohn's work. Precipitation is a much simpler process in principle, and it's low cost, low process mass indices. It's high throughput, eminently scalable, and it's very low complexity. So it addresses a lot of the sustainability concerns that we have with current platform processes. All right, so how are we going to generate this precipitate? Well, it turns out there are a number of different ways you can precipitate antibodies, but it turns out that there is a combination of zinc ions and biethylene glycol 3350. Those of you of a certain age will recognize polyethylene glycol 3350, having drunk probably several liters of this material before your last colonoscopy. So zinc and PEG together, pretty selective for antibodies. The zinc will bind to histidine side chains, imidazole residues of histidines, and form a crosslink. And I've got that shown here up in the upper right-hand corner. So that's the Y-shaped molecule, and zinc cross-linking sites can link those together. They're reversible. You can drop the pH, and protons will outcompete the zinc for the histidine residues. Zinc will bind to other places besides histidine, but histidine is the favorite. Polyethylene glycol operates by a completely different mechanism. It's a volume excluder. These are 150,000 molecular weight molecules. They're big. Most of the other contaminants are small. High molecular weight stuff, okay, right? But there is a lot of stuff that's smaller. So the PEG is going to exert more of an effect on these big guys, like the antibodies, than on the small guys. In combination, they're very effective and relatively selective. Are they as selective as protein A? No. Do they need to be? No. So we understand, we think pretty well, the action of this precipitant combination. As a matter of fact, we've got some detailed solubility modeling that describes in the colloidal sense the various dependencies on pH, ionic strength, and you name it. I won't talk about that here today, although I would love to. We have characterized the solubility behavior of many antibodies in this PEG and zinc system. I'll show these heat maps. I know they're kind of small and squinty, but just look at the pretty colors. So the idea here is I've got zinc on the y-axis and PEG on the x-axis, and the colder colors are lower solubilities. The idea is we need to add enough PEG and zinc to knock down a good amount of the antibody from the solution. We typically like to see at least 95% yield, and these dashed lines here indicate where the 95% yield lines are. More PEG, more zinc, more precipitant. We notice that there are some patterns here. There are a number of mAbs that have this codependence between zinc and PEG. These have kind of L-shaped, we have a lot of corners in their solubility curves, so there's synergy between the two precipitants. And we also notice there's a class of antibodies where it's mainly zinc that exerts its effect. We still use PEG because PEG helps us tailor particle size distribution, but you can see the behavior is very different. So this is for this first column on the left is for purified mAbs. When we look at mAbs in HCCF in crude form, solubility behavior is a lot different. And this was a reckoning that we had to deal with. And it turns out that if we do some diafiltration, if we remove residual phosphate, which competes for zinc in solution and phosphate's present in cell culture fluids, then the behaviors we get in diafiltered or dialyzed crude antibody mixtures pretty much recovers the purified protein behavior. That was an important finding. We've done dialysis here with 3 kilodaltons molecularly cut off filters, so we know it's a small component, and we're pretty sure it's phosphate that is the bad actor. All right. And we've noticed some common themes here. So a number of antibodies we've used here, I'm showing 7 different antibodies here. Whether or not the precipitation is zinc and PEG codependent, kind of synergistic or just zinc dependent, seems to break down based on the isoelectric point of the protein. That's the pH at which it has net zero charge. So it kind of stands to reason, if you're trying to drive things together and cross-link them, if they, in a colloidal sense, have relatively low charge, pretty easy to do. If they're highly charged, you got electrostatic repulsions to overcome, right? You're thinking about these as charged spheres, right? So there's more to overcome. Hence, PEG helps drive things together, provides a driving force from an excluded volume sense. So this was an important finding. I show this data in Heap's buffer, which is a non-interacting buffer. It has very low interactions with zinc ions. Situation can change a little bit if you have something that competes with histidine for zinc. And Tris buffers, Tris acts like a weak ligand for zinc, and it tweaks behavior a little bit, but I won't focus on that today. All right, so we've done some solubility screening before we realized that dialysis was really helpful. We figured out, okay, what are the conditions that'll generate 95% recovery? They're kind of all over the map, right? 8 millimolar zinc and 6% PEG, 15 millimolar zinc and 7.5% PEG, and we didn't even get to 95% recovery here from that too. All over the place. After dialysis and removal of the small components, everybody's pretty much in the same region, and very little zinc, 3 millimolar, very little PEG, right? 3%, 1.5%, 2%. So we know where to look now. If we pre-process our cell culture fluids with dialysis, if you're going to do dialysis, might as well do diafiltration with a bigger membrane and eliminate some of the smaller contaminants as well, smaller host cell proteins, et cetera. We're in this small corner of the solubility diagram, so we know where to look. We can tune this thing. We do get a reasonable purification. I'm showing down here, this is the size exclusion, set of size exclusion hematocrit. So the separation is based on size in this media. Monoclonal antibody here, this is for mAb3 in particular. And we take care either to inject the equivalent amount of mAb in every sample or else normalize our chromatograms in terms of the peak area of the mAb. So we've got multiple chromatograms overlaid. You can see the reproducibility mAb to mAb is very high. This allows us by inspection now to see how we're doing purity-wise, because the area under the peak is an indication of how much material is present in that mixture. So same amount of mAb present all the time. Our crude raw cell fluids in this black dashed line. Some pre-processing, change the distribution a little bit. After precipitation, it's down here in these little lines. So I got to blow it up. And if I blow it up, you see the two lines up top that blow up through the top of the diagram. You know, they're too concentrated even to plot on this scale. But now if I focus down, I can see what's left. And I can tweak the conditions. Here, 2% PEG and 2 millimolar zinc. And I could say, well, if I want a higher yield, what if I boost the concentrations of the precipitants? Well, the yield goes up, but purity suffers. There's a yield-purity trade-off, right, as you'd expect, because now if you add more precipitant, more other things are starting to get included in the precipitant phase. So you'd want to operate under conditions where you meet your yield target, but really no stronger conditions than that. If we go over to the left-hand side here, where big things are, in size exclusion, big things come out before small things, right? Because big things can't get into the pores and the small things spend time in the pores, as they transit the column. You can see this collection of curves that indicate the high molecular weight species that are present. And we don't really do much in terms of purification of high molecular weight species. We get a great benefit for the low molecular weight species, but don't make too much of a dent. There's a small dent in the high molecular weight, but it's mostly low molecular weight. So we're going to count on the polishing steps to get rid of any residual materials. We've knocked it down tremendously here in precipitation. We've got more to get rid of, and we've got to do some work on the high molecular material. So we'll rely on our polishing steps for that. And for polishing, we're going to do chromatography. We're going to do a flow-through mode of chromatography here. And the idea is that because the precipitation operates well in a continuous mode, let's try to make the whole process continuous, because we get benefits in terms of throughput from operating continuously. And if we do a so-called, subtractive mode of chromatography, we're going to remove all the trace impurities and let our mAb sail through without binding. There's a lot of different resins we could use. There's certainly lots of guidance from the literature on the platform process about which resins are good for polishing purification. We know that typically some hydrophobic resin would be a good thing to use, and we know that some anion exchanger would also be a good thing to use because antibodies are going to be positively charged, and an anion exchanger is positively charged, so antibodies are not going to want to bind to that. Pretty much everything else does like to bind those things. We do some screening in plates to figure out first who binds and who doesn't, and then go to mini columns to figure out, okay, based on things that look promising in plates, now when you start to blend in transport properties in small columns, does that still stand up? We get some capacity information, relative capacity information, and then we can go from that screening to select a two-step process for the polish. So our first polishing step, we mentioned that hydrophobic media are a good thing to use, and it turns out activated carbon is like a magic bullet. So we tried activated carbon, and it worked wonderfully, and it was like full stop. Do we even need to talk about anything else hydrophobic? It works so well. So here, another size exclusion chromatograph. Here's the mAb feed. Now we're looking at blowing up this region. So here's redissolved precipitate, right? Tiny little tiny peaks, and now we're making them even smaller. We can tweak the conditions. We figured out that lower ionic strength conditions, so lower strength buffers, works the best, and we can drive this down to really small levels. I didn't blow up the high molecular weight area because the hydrophobic separation doesn't do much for the high molecular weight region. It's the low molecular weight materials that are attached here. So we've got a magic bullet here that works really well. For the second polishing step, so our orthogonal mode, we looked at mixed mode anion exchangers. So these are anion exchangers that are positively charged species that have a little bit of hydrophobicity with them too. And there are a number of these different resins. So we took a selection of popular ones and did some screening in a plate basin. And we screened different pHs and salt concentrations to see Where are conditions where the monoclonal antibody flows through? We've got to figure that out first, because if it doesn't flow through, there's no point. Right? So where it doesn't bind, and the green colors are friendly regions, the red colors are unfriendly regions. Then, having identified that, take a look at what happens to the impurities. Where do they go? What's the capacity for binding? How much is removed? And here again, the red areas are where, well, we don't do such a great job of removing, and the greener areas are where we do a better job removing. And we looked at both low molecular weight species and high molecular weight species. And you can see the high molecular weight species in particular are knocked down by this mixed mode approach. So we can find conditions where we do this dual optimization, right? Where we get high yield and also have good binding capacity for the impurities. And this is just showing another chromatogram, size exclusion chromatogram, of how we do a number of different media under conditions that work really well. And you can see we make a little bit more of a dent in the low molecular weight species, probably down to very small levels, and we take a big bite out of crime here with the big guys, right? So we've got conditions, right, and media for precipitation and for the polish. These all work, right? So let's make a process. And this is linking the polishing steps together, so how we go from one step to another. And another underlying requirement that we have is, can we do this in a way where we minimally manipulate the solution conditions, right? We don't want to have to do a lot of pH adjusting and salt adjusting, et cetera. We have to do some, but it turns out we can go right from redissolved precipitate onto the activated carbon column. And then after that, we do have to do some pH adjust and ionic strength adjust to get to the second column. And we're working on trying to minimize that as well. So we've got processes for different antibodies that look very similar. And we have good yield and purity results. These are just numbers corresponding to those diagrams that I showed. Actually, these are from mini column experiments to show how we're doing on yield. We have great yields through this process, and how we do the clearance of host cell proteins and high molecular weight species. So in terms of parts per million, right, we're getting down after the second operation to really small numbers here for host cell proteins and for the high molecular weight species. All right, so we put this all together in a process. So here's the process, right? Pre-processing. DNA depletion. So I didn't talk too much about this, but we find that if we knock down the DNA a bit up front, it helps us out later on. So we do a calcium chloride precipitation to get rid of DNA. Calcium chloride forms calcium phosphate with the phosphate ions in the host cell culture fluid, and it's the calcium phosphate that's the actual precipitate for DNA. So we knock down phosphate too at the same time. We'll also do some concentration diafiltration, because here's an opportunity to knock out any residual phosphate, but we can also pre-concentrate. And it turns out if we pre-concentrate, performance improves too. All these performance metrics that I mentioned are per gram mAb. So if we concentrate and operate at higher mAb concentrations, all these per gram metrics look a lot better. And this also allows us to compensate for the fact that, oh, one particular bioreactor run had this tighter concentration of mAb, another run had this different type. We can adjust to get to the same point so that the feed looks similar every time. Once we pre-concentrated, start over here. Precipitation, we've got a two-step precipitation. Turns out we use zinc first to make nuclei, and we drive them together by adding PEG to make particles that have an appropriate particle size distribution and morphology. Our next step is dewatering. We get rid of the supernatants, right? We selectively precipitate the mAb, so everything we don't want presumably is left behind, so we try to remove as much of that as possible. We do this in a hollow fiber filter module. So we pass precipitates through a hollow fiber system, which you would think would be a crazy thing to do, because you're going to plug it and it'll be miserable. If you've got good particle morphology, it works. If you don't, it plugs and it's miserable. Two-step washing, counter-current washing, where we add in fresh buffer to remove any residual supernatants. And we can do that. One of the important metrics here is the conversion. So what percentage of the material that comes in exits with the permeate? The more material that exits with the permeate, the higher the purification, right? The more material that we're taking out that's not part of the precipitate phase. but the slurry becomes more concentrated and harder to filter, and it gets easier to foul the filter. So we've got to be careful here in adjusting the morphology of the precipitate so we can get high conversions. All right, two stages, counter current, saves on solvent use, so saves on buffer use. Then we go to a redissolution step, we just drop the pH. Turns out that viral inactivation in the platform process, we hold the pH at 3.5 for about half an hour. This precipitate redissolves to about pH 5.5. It'll redissolve at 3.5 too, so you can hold it there. So this is a nice drop-in step that also accomplishes the viral inactivation. We connect this to a flow-through polishing step, activated carbon, and a second flow-through polishing step. Connecting these steps because we have the outlet of one pump pumping into the inlet of a second pump. So we're essentially deadheading the first pump, like there's a second positive displacement pump. How do you do that without blowing up a tubing, right? We've come up with these flow attenuation modules, just another hollow fiber filter repurpose where we purposely set the flow rate coming in higher than the flow rate going out. We have some permeate flow, and this permeate flow can fluctuate to match the flow differences and fluctuations between the pumps. So it simply is. So that's what the process looks like. Here's what it looks like on the lab bench in 4432 over at CVIS. So here is the zinc-based precipitation. Here's the PEG-based precipitation. These are tubings with static mixers in them, a little bit more residence time to develop the appropriate morphology after the PEG is introduced. We have a series here. Here's our watering hollow fiber filter. Here's the two-stage counter current. Further precipitate washing system. Here, redissolution of antibody. Here's the first chromatography step, flow through chromatography step. And then, we got the configuration lab, long tubing, the second chromatography step over here with a mixed mode anion exchanger. And the cool thing here, 30 minutes from there, molecule enters to molecule becomes bulk drug substance. Now, this hasn't gone through a formal viral inactivation step, and it hasn't gone through a UFDF step prior to formulation and fill, and there's a viral filtration step too. But all of that together for the platform process is typically several days. 30 minutes. Pretty good. All right. Geez, I'd love to talk about this all day, but I better not. So let me move expeditiously. So I'll talk about the performance of this process. So the pre-processing, the whole point is to get rid of the phosphate, get rid of the DNA, knock it down to a reasonable level. We do a good job of this. The DNA clearance reduces headaches later on. As we redissolve later on, if there's a lot of DNA present, at some point we're going to neutralize the solution. We'll pass through a pH that's intermediate between the isoelectric point of DNA and the isoelectric point of the antibody. There'll be an electrostatic attraction and there'll be a precipitation. A few companies actually use this to remove DNA, so they sacrifice some of their monoclonal antibody to remove DNA. We could do this, we chose not to, right? And so we're removing the DNA up front. And then down here, as I mentioned, the concentration diafiltration step, this sets a desired constant concentration for the downstream process, and it does clear any residual phosphate, so it's not competing with the zinc. And we can use a bigger pore membrane here. If we're going to do dialysis at 3,000 molecular weight, why not do diafiltration at 30,000 molecular weight cutoff and remove some of the small impurities too? And so we get a modest removal of smaller impurities. This is the heart of the capture step. So the precipitation, the two-stage precipitation, dewatering, and then the two-stage countercurrent wash. But protein A sets the benchmark. Now we don't have any illusions that we're going to perform as well as protein A does. Protein A yields are typically greater than 95%, maybe around 98%. Great post-cell protein clearance, 2 orders of magnitude. Throughput, good. Buffer consumption, about 500 milliliters or so per gram mAb. In our work with this, our yield was high. Post-cell protein clearance, not so great. So it's going to be hard to compete with protein A on that, but we don't need to, as it turns out. We need to be good enough. Throughput, numbers, they look okay. The throughput numbers on the filters look really good. The buffer consumption is down quite a bit, and that's promising. The key thing is the conversion. So finding particle morphologies, and we have an online particle monitor that will give us information and images of the particle morphology. And we're working on capturing this information so that we can use it in closed loop control of the precipitation operation, so we can dial in the morphology and keep it there during the course of a process. Right now, we fly by experience, and we can tell by looking at the size distribution and the images about what morphologies are reasonable. Conversion is key in the performance here. So if our conversions are low, 40 or 50%, we don't get good purification. If the conversions are high, we get wonderful purification. And as the conversions increase, every percent, it's a geometric response. So every percent increase in conversion, we get an even greater gain in purity. And we figured out that reducing host cell proteins to about 20,000 parts per million, even though protein A can typically get down to several thousand, it's good enough because the flow-through polish steps are so good at removing residual materials. If you have more than 20,000 parts per million, what the problem becomes is now you've got so much impurity present that you're really taxing the polishing step, and you're loading it up, you're dunking it up with contaminants really quickly, and you're going to have to flush and clean those columns much more frequently. So if we're down in this range or lower, we have good performance for the polish. First polish step is the activated carbon. And here we get nice yields. So we were aiming for at least 90%. We're close to 95%. Great removal of host cell proteins, right? Over 2 orders of magnitude. Throughput is really high. As I mentioned, low pH Rita solution gives easy insertion of an inactivation step, a viral inactivation step if we want. A little novel flow attenuation module, which means we operate fully continuously without surge tanks. Right? Why not a surge tank? And activated carbon is like a magic bullet. There are other issues with it, right? But activated carbon works really well. And this permeate that comes from the flow attenuation module, it's a pretty pure solution. We may be able to recycle that for use further up in the process where the system has more contaminants. The second polish operation, again, yields about 90% or better. High throughput, or the HCP clearance, right? Pretty good. It's got great clearance of high molecular weight species, good throughput, good capacity. And again, we have opportunity perhaps for reuse of the fluids coming out of the flow attenuation module. And again, we don't need a surge tank. We did have a headache because we have a little bit of residual zinc that we need to remove. And here, when we reneutralize, if there's a little bit of zinc around, well, we precipitate under neutral conditions with zinc. And so we can get formation of a little bit of fuzz, right? A little bit of precipitate. So we've got some residual zinc that we've got to deal with. All right. I'm going to walk through that, just boom, and go to this bottom line. So what does this all mean, right? We have these big sustainability goals. So how are we doing? So a number of metrics here over on the left. Yield, right? We've got to have good yield. We don't have good yield, there's nothing to write home about. We want to reduce buffer use. We want to take a look at, what is our throughput of culture fluids, right? For capacity. What is our overall yield? What are the host cell protein levels we can achieve with this process? How about the DNA and high molecular weight species levels? And what's our cost of goods? So here are targets based on the best-in-class periodic continuous protein A platform for purification of mAbs. So we should be 85% yield or better, buffer usage below 1 liter per gram. Throughputs here about 26 grams per liter per hour at 5 gram per liter titer. Overall yield, capture and polish, better than 70%, 75%, 100 parts per million on HCPs, less than 10 parts per billion on DNA, less than 1% of high molecular weight or aggregate species present. And calculation was done at the University College London that says this process, best in class, about $63 a gram. Right? That's their prediction. Same group did an analysis of our first hack at this from 2019. So this is the first work that we did in the area. Show our metrics. Our metrics weren't bad, right? And they said, okay, $67 per gram, so you're in the same ballpark of the best protein A-based process. Okay, cool, right? We can build on that. So work that we did, subsequent work, we didn't have a cost analysis here, but we did really well in terms of capture yield, buffer usage, our throughput is up. Our yield, our total yield, 65% needs to be better. Our purities were excellent. And most recently, we ran the whole process continuously for, it was about 4 or 5 hours. We had everything linked together, everything operating continuously. 96% yields, right? Our buffer usage down 0.355 liters per gram. Our throughput looks pretty good. The overall yield, 83%. Low as 3 parts per million for the host cell proteins. And the DNA that was here in the similar process was below our level of detection, so we didn't use it. We're using our crude detection technique, and we need to step up and use PCR. And our host cell, or our aggregate down below 1%. Sartorius estimated, did an independent analysis incorporating some of the changes we made relative to the analysis that Nevis et al. Did in 2024. Not everything, but some. They're predicting right now we're at $34 a gram. We're on our way. We're on our way. We're going to get there. All right, so let me summarize here. So we figured out solubility behaviors in the system, really exciting. We got a lot of really exciting work in modeling the solubility here in a collaboration with Patrick Underhill. And the mAb PI, it was really cool finding that it's the PI that seems to determine classes of behavior in this 2-precipitant system. And the removal of small interfering components was key in order to achieve the use of small levels, low levels of these precipitants to generate the mAb precipitate phase. The performance here of the membrane operations, right? It's critically important, and it depends on the morphology of the precipitate. We know kind of by the seat of our pants how to get there. We're doing some modeling work, population balance models, to figure out how we can dial this in and couple it with our online measurements for particle imaging to actually control the behavior of these precipitators. The resin screening, that was very efficient, made very efficient processes, even when we had impurity levels as high as 20,000 parts per million of host cell proteins. It activated carbon. It's a magic bullet for host cell proteins, which is wonderful. And we got really nice performance of this pairing of operations. And we hit nearly all of our performance targets. Nothing is optimized here, right? We just found conditions that worked and ran with them. And we're doing really well so far. And now that we're in an optimization phase, we're hoping that we're going to make even greater strides. One of the ways we're going to do that is by intensified operation, meaning we'll operate at even higher protein concentrations. Again, everything scales per gram. So if instead of operating at 3 grams per liter, we operate at 10 or 25 or 30, right? All of those metrics look better immediately. Are there opportunities for improvement? Absolutely, and we're working on that now. All right, so here's the team. I want to thank collaborators, Steve and Wayne in particular on the process, Andrew Zidney at Penn State, whole host of students and postdocs, those that have their doctors in parentheses, got them while they were working on the process as part of their work. And I want to call out Matt, Mirko, Ali, Mariusz, Olivia, Mario, Hannah, Andreas, Jacinta, and 2 new members here that are being trained in the process. They're on the quest for $10 a gram, Sunik and Manas. Here's an earlier shot of the team that's from a couple years ago and had Ali and Mirko in it, which is why I have that included, part of the group. I have this little inset here. Before I go to that little inset, I want to acknowledge Nimble for support, the Food and Drug Administration, and the Gates Foundation for supporting us on this quest. And I mean, I feel pretty passionate about this. And I could talk forever on this, and I know you need to get on to the rest of your day. But it's something I feel very passionate about, and it has a way of rubbing off on people. So this is Mirko from Penn State. He spent a week with us. He spent a lot of time with us, more than 1 week. But during one particular campaign and run of operations, he spent a week with us. He's a Penn State graduate student. Oh, look at him. We converted him. RPI has that effect on folks. All right. So at this point, I'd be happy to take questions. Yeah.
I'm just so what do you think, what is the toughest quality attribute in like during precipitation for like fragmentation, aggregation, or charge?
No, that's a good question because, certainly aggregates are an impurity that we need to remove. You might be thinking, oh, aggregates, well, why are you precipitating? You must be generating a mess of aggregates. Turns out that the formation of stability aggregates different than these cross-linked aggregates. So it's not really the same species. We don't tend to remove aggregates on precipitation, nor do we generate more. So they're kind of unaffected and they go along for the ride in the precipitate phase. And it's predominantly the last flow through chromatography step that knocks those down. One of the things that we think about the most is the host cell proteins during the precipitation. So that's like a key attribute. We know we can get yields, right? Because we can always add more precipitant, but that purity suffers. And in order to be reasonable to the downstream, the further downstream polishing operations, we got to knock those cell proteins down below 20,000. If we knock them down to 10,000 or 7,500, that would be even better. So if we can put pressure on that, that'll help the polishing operation perform even better than it does. So it's kind of those two things that
It's Todd. It's amazing the progress you've made. And I just wanted to ask, just to put it all in perspective, like when did you start working on the project? I think you mentioned 2019 was like one of the first iterations.
yeah.
So like 2018, 2017?
So the idea was hatched at a recovery conference in Rostock, and that was 2014. And it took 3 rounds of knocking on NSF's door before we finally got in the door to get some initial support. And when that happened, we ended up starting a project in about 2016. And that led to the work that was published in 2019 with Andrew Sidney and my prior academic institution. And we brought that project here, and then it kind of blew up. During COVID, right, folks realized that, hey, manufacturing of biotherapeutics, geez, it's become really important. And so there was a lot of opportunity for funding. In parallel, Nimble came on the scene. And so the support for bioprocess research, which for a lot of times you knock on an NSF door, don't even bother on NIH's door because it's like, oh, this is plumbing, it's not so interesting. Go do some metabolic engineering or something else. So interest came back around when folks realized, hey, making these things and having an adequate supply, that's important. At some level, it doesn't matter, all the wonderful engineering and science that's done upstream to make a drug if you can't, or to find a drug, if you can't get it out to everybody. So that has helped quite a bit.
I think this is a great perspective, especially for like the younger students, even the junior faculty, that like progress like this takes a decade of sustained effort, multiple PhD projects, and it all adds up to this, right? So I think any of us who are like thinking, oh, haven't made a lot of progress in the last year. I mean, it's like a long game, right?
Yeah, we're standing on the work. You see the list of people that have a major part in the process. We wouldn't be where we are without the whole team. And I will point out too that there is the the classic trade-off between the true believer that I know this is going to work, I know it's going to work, and when do you say, well, okay, I gave it the good college try, and I need to do something different. So that's something you have to address in each of your research projects, right? What, this has got to work, it's got to work, it's got to work, I know it's going to work. And then, realizing that sometimes research hands you no for an answer and you've got to change. So we were dogged, like a dog on the bone in this, moving this forward, particularly early on with NSF, keep on hammering on their door. We know this can do it, we know this can do it. Gates Foundation believes that we can do it this way. That's really cool. But you do have to, again, keep your eye open. Is my deep belief in this, is it clouding my vision to the scientific realities? So that's a struggle we all have.
Yes.
Yeah, I was just going to say that another comment here is also supply chain issues. So if one of those Alzheimer drugs actually hits, right, there won't be enough protein A resin in the planet to do the process, right? So I think the real advantage of this would really be seen when you go to a much, much larger scale. And then there might not be many options, right?
There's a secondary supply chain issue that, I mean, supply chain is part of sustainability, but you know, if you're going to manufacture drugs globally, a lot of countries will say, look, these drugs are expensive, even if you're selling them at reduced cost. And the reason, again, $10 a gram is we need to get to that cost to be able to sell it at $10 a gram to the place that need it, that can't afford now. So that's got to be the selling price, right? The idea that a lot of these countries are saying, look, it's expensive for us to buy these drugs externally. We want to capture some of that value chain. We want you to do some manufacturing here, right? In place. So we capture some of that value. So our people are employed, et cetera. So we have the supply locally. And so simplifying the supply chain and simplifying the process, not because they can't handle the complexity, there are smart people everywhere. It's that getting these complex needs to these sometimes very remote sites is a challenge. So if you can do things more simply, then more places will be able to do the manufacturing.
And John should help us start a company to do that.
I'm going to use the George Belfort question and comment approach. So going back to your 2 a.m. commercial infomercial thing. So when you see these, it drives me insane because, you know, 95% of the time it talks about all the side effects, right? And so we've been through this process. So the whole problem is upfront, before we even get to the manufacturing, is the design of the mAb to begin with. You know, what antigen are you tackling? If this antigen is on both the unhealthy cancer cells as well as a healthy cell, then that can lead to autoimmune diseases and all this kind of stuff. So that's kind of the stuff. So just that's the comment. So my question to you is the upfront show part. So now you've got, we can do 30 minutes downstream processing, right? But still, if we use like a fed batch reactor upfront, we're still at the 4 or 5 days, that sort of thing. Any comments? Yeah, maybe longer. Any comments about what we need to do upstream to speed up the overall manufacturing?
Well, to be honest, it's the downstream that needs to catch up to the upstream. There was a pretty influential person who I love dearly in downstream processing, National Academy member, but who has made the statement in published work that says, why bother improving the upstream manufacturing because downstream can't keep up with it. So there's no point in going above, say, 6 grams per liter in the upstream titer because the downstream can't handle it. So it's interesting. So it really brings out the fact that it's the downstream that's the limitation right now. Moving to perfusion is helpful in terms of throughput. It uses a lot of raw material, so being able to perfuse at lower rates is important because the media usage is really high and the titers aren't as high. So the concentrations of antibodies in the perfusate lower, significantly lower than you get in a fed batch. But we have the pre-processing step. In the pre-processing step, we can dial in essentially whatever pre-concentrated level would be helpful.
You had shown on that Sartorius one that it broke down about $34 a gram. How does that break down between upstream and downstream, number one, and number two? How does the cost of goods relate to the overall process cost?
So the cost of goods is the overall process cost, right? Through bulk drug substance, right?
No, but I mean, from a planned depreciation, the whole bit.
Oh, well, cost of goods doesn't include that. So it's a net present value calculation or an ROI calculation that would treat that. The first part of the question was?
The breakdown between the.
Oh, breakdown. It's like seat of the pants. It's roughly 60% of the cost of downstream costs. Numbers have been cited anywhere between 50 to 70%, about 60. So the bulk of the costs are in the downstream. And, right.
Of all the steps in your purification scheme, which is the hardest one to scale up for industrial production, and what's the reason?
That's a good question. You know, the precipitation, it turns out, isn't hard to scale up. Once you have the conditions dialed in at small scale, you match the power input per unit volume for the mixing, and you'll get the same particle size distribution. We showed that in earlier work where we scaled a range. We scaled down a factor of 10 and up a factor of 100. Match the particle size distribution. It's finding that particle size distribution in that morphology initially that's the challenge. Once you find one that works, okay, we can replicate it. So that's really not too hard. And even the flow-through chromatography, once you've found conditions that work well, you just have to have enough residence time to capture the impurities before they flow out of the column. And it's one of the simplest modes of chromatography because it's just running continuously and you're just loading up the column with contaminants. Once they start to break through, okay, stop, switch to a new column, let's take that old column and let's regenerate it and get it ready to use again. So you can switch back and forth. So the scale up is really not a challenge from either perspective. So we're fortunate there. It's a simple enough process. That's what we try to hammer on the complexity. I don't have a good measure of complexity though, for the comparison.
Maybe one last question.
Yeah, I think talk about this stuff all day. Thank you for your.
Yeah, George, did you want to ask a quick question?
Sure, it's never quick. I do have a question. May I go now? Todd?
Yes.
Please.
Terrific talk. I loved it. I have a few fundamental questions. The first one has to do with, I remember Maria Cooler was worried about the cost of PEG when she did two-phase aqueous 20, 30 years ago. And so she did something very clever. She actually was able to show you didn't have to purify the PEG. You could use crude PEG. And it didn't work 100% equally, but a little bit less. So there's a possibility that if you could look at crude PEG and not use the purified, you'd save a lot of money. So that was one suggestion. I thought you would say something about two-phase aqueous because you can get, of course, RNA to go in the top and protein to go in the bottom, and you can actually separate them. Did you consider two-phase aqueous at all in your process?
No, we didn't because, two-phase for protein separation has been kicked around for a long time and it really hasn't broken through. I don't think it could compete as well in this case. Unlike for the RNA case where the two-phase works remarkably well. So I think, the precipitation seems to work a lot better. And again, there's the historical precedent from Kahn's work that showed that, hey, this is viable. It's been used on commercial scales since World War II. Regarding the PEG comment, so we use PEG 3350. It's small, it's easy to remove. It's generally regarded as safe. Again, you drink liters of it if you're getting ready for a colonoscopy. It's in your future, right? Not something to look forward to either. So that was our primary choice there. And the fact that we did the dialysis meant that we removed the small interfering components, we can get away with using a lot less. So whereas an aqueous two-phase system might use anywhere between 16 to 25% solution of PEG, we're using 2% solution. So we're down at a level where our usage is minimum. And that's a good thing too, because PEG is our biggest greenhouse gas generating component in the process. So.
All right, because the time was.
One last question, just the last one.
Gotta be fast because people need to get on to their day.
It's a philosophical question, and that is you've come up with a whole process to replace protein A. Do you think one could come up with an actual process to replace protein A with a different capture? Say, for example, a peptide, or what about using, what do you call those? Not peptides, but there are other ways to also.
Yeah, other affinity groups, aptamers, et cetera. Well,
aptamers, yeah, I was thinking of aptamers.
So, I mean, the classic route to biopurification is affinity first and then cleanup, right? And so there are lots of alternatives because of protein A's high cost. There's a lot of market opportunities for folks that can come up with something that can compete with it that are costly, but not nearly as costly. They're all viable choices. What's driving our use of this kind of, we'll say, lower tech approach is just the fact that it is lower tech. If, as we saw during pandemic times, supply chain issues become a real problem. Access to PEG and zinc may be a lot more convenient than access to protein A or specialized peptide, that sort of thing. So we're trying to go a lower tech route that would be, applicable globally.
All right.
Well,
Thanks
very
a lot,
nice.
Todd.
Thank you.